Hello healthcare viewers, this is Gaston, admin of the healthcare engineering team. As I have explained in the 3D bioprinters part 1 video, we are releasing part 2 which is continuously giving information about 3D bioprinters. So we would like to request our healthcare viewers and we request you to share this video with your colleagues. We request our healthcare viewers to watch this part as well. If you miss our part 1, please watch part 1 of the 3D bioprinters and get updated your knowledge. We kindly request our healthcare viewers to subscribe and share to support our healthcare engineering team, and we promise that in future we will release so many informative videos similar to these to update your knowledge. Thank you. Keep watching.
First thing is print head mount. On the bioprinter, the print heads are attached to a metal plate running along a horizontal track. The x-axis motor propels the metal plate, that is a print heads, from side to side, allowing material to be deposited in either a horizontal direction. Next one is elevator. The metal track running vertically at the back of the machine. So this basic process of an elevator is to moves a print head up and down, and always the elevator is driven by the z-axis motor, so it makes it possible to stack successive layers of material on one top of the next. So this is the basic process. Next one is platform. A shelf at the bottom of the machine provides a platform for the organ to resting on during the production process. So the platform may support a scaffold and petri dishes and also a well plate, which could contain up to 24 small depressions to hold organ tissues samples for pharmaceutical testing. A third motor moves the platform from the back along the y-axis. Next one is reservoirs. So reservoir is a common thing in the basic printer and also in the 3D printer. The reservoir is attached to the front head and hold the biomaterial to be deposited during the printing process. These are equivalent to the cartridges in your inkjet printer, as we know before. Next is print head syringes. A pump forces material from the reservoirs down through a small nozzle or a syringe, so which is positioned just above the platform. As the material is extruded, it forms the layer on the platform. Next is triangulation sensor. A small sensor tracks the tip of each print head as it moves along the X, Y and Z axis. So it acts in both X, Y and also Z axis, so that it senses all the axes. So it communicates to the software with the machine, so the precise location of the print head is known throughout the process. This is the basic process of what a triangulation sensor do. Next is microgel. Unlike the normal printer ink at home, Bioink is alive, so it needs food, water and oxygen to survive. So because it is a cell, right? So this nurturing environment is provided by a microgel. Think like gelatin is enriched with vitamins, proteins and other life-sustaining compounds. Researchers either mix cells with the gel before printing or extrude the cells from the one print head, microgel from the other. Either way, the gel helps to cell stay suspended and prevents from setting and clumping. So this is the basic process going on for the users of microgel. Actually, it acts as a medium for doing something. Next is Bioink. For Bioink, organs are made of tissues and tissues are made of cells. So this is the basic concepts or we are all know. To print an organ, a scientist must be able to deposit cells specific to the organ, so they hope to build. For example, if we want to create a liver, we should start with hepatocytes, the essential cells of the liver, as well as other supporting cells that related to the hepatocytes, that's the cell of the liver. These cells from the special material known as Bioink, which is placed as a reservoir of the printer and then extruded through the print head, as well as the cells accumulate on the platform and become embedded in the microgel. They assume as a 3-dimensional shape that resembles the human organ. So this is the basic components and their functions that present in the 3D bioprinter. Basic steps to print a complex organ. Always there is some basic steps needed for every action. So for printing a complex organ, there is a basic steps needed. First thing is we have to take a scan, or the doctors make CT or MRI scan of the desired organ. Next, they load the images into the computer and build the corresponding 3D blueprint of the structure using CAD software. Combining this 3D data with histological information collected from years of microscopic analysis of tissues, scientists can build some slice by slice model of the patient's organ. whatever the organ may be, maybe a liver or a heart or whatever. Each slice accurately reflects how the unique cells and the surrounding cellular matrix fit together in 3D space. After that, it's a matter of hitting file, print, which sends the modeling data to the bioprinter. The printer outputs the organ one layer at a time with the help of the Bioink and gel so that it clears some multicellular complex tissue and hold it up in the place. Finally, scientists remove the organ from the printer and place it in an incubator, where the cells in the incubator and the Bioink enjoy some warmth, quiet downtime to start living and working together. So, for example, we take, as we discussed below, or before, the living cells need to form what's biological called tight conjunctions or the junctions, which describes how the cell membranes of one cell fuses the cell membranes of adjacent cells, so that we will get a clear matrix that will be created with a perfect manner. So the time in the incubator relay pay off. A few hours in the warmth turns the Bioink into living tissue capable of carrying out liver cells, and also surviving in a lab for up to 40 days. So the final step of this process, so this is the main step, that we are going to print an organ that it behaves like a native cell. So it's really been challenging. So this is the basic steps that needed to print a complex organ. Bioprinting process for printing organ. So once the tissue's design is established, the first step is to develop the bioprocess protocols required to generate the multicellular building blocks, and also it is called as a Bioink from the cells that will be used to build the target tissue. So the Bioink building blocks are then dispensed from the bioprinter using the layer by layer approach, as we studied above. And also the bioprinting process can be tailored to produce tissues in the variety of formats, from the microscale tissues contained in standard multi-well tissue culture plates to larger structures suitable for placement in the bioreactor prior to use. Next is bioprinting process for printing 3D models. To make a 3D model using this technique, a manufacturer loads a substance, usually plastic, into the mini fridge sized machine. So we also loads a 3D design of model we wants to make. So when he tells the machine to print, it heats up, and using the design as a set of instruction, extrudes a layer of melted plastic through the nozzle onto the platform. As the plastic cools, so it begins to solidify. Although it by itself it becomes solidified, it's nothing more than a single slice of desired object. The platform then moves downward, so a second layer can be deposited on the first. So this is how it works. The printer repeats this process until it forms some solid object in the shape of certain 3D model we are going to design. This is about the basic steps for 3D models. Bioprinting implication. So risk of transplant organ rejection should be very low. So it acts as a pursuit of life extension, and also bioprinting increasingly learning to manipulate living tissue at its cellular level. This solved problem. So rejected ones, it transplanted in patient body. So they use a 3D scaffold made at biodegradable polymers or collagen. So for a simple organ such as bladders, researchers don't print the living tissue directly. So instead of that, they print a 3D scaffold first to make a biological or a biodegradable polymers or collagen. So determine the exact shape of the scaffold, they must build a 3D model with the help of the CAD software, so computer aided design. They usually define the exact X, Y and Z coordinates of the model by taking different scans like CT, MRI of the patient using the advanced technology, as we said before, that computerized tomography or the magnetic resonance imaging. So to produce the above works, become a bladder tissue only contains two types of cells. So organs like kidney and liver have a far more complex structure with a greater diversity of cell types. So while it should be easy enough to print a scaffold, it would be almost impossible to recreate the three-dimensional structure of the tissue manually. A bioprinter, however, is ideally suited to complex such as time-consuming and detailed-oriented tasks. The important thing is that we have to able to print accurate adjacent matrix so that it forms a structure that we have native cells perform, and also the functions related to that. So this is about the solved problems in bioprinter. Uses for 3D organs. So replacing part of the skeleton is one area being revolutionized by 3D printing. So the printing entire bone for the placement in the body. Doctors in the Netherlands have already created a lower mandible on the 3D printer and implanted the jaw made from the bioceramic coated titanium. So in the patient suffering from a chronic bone infections like that. So next one is, some dentists now take an intraoral scan of the patient's teeth and send the scan to the lab that fashions a proscenium bridge using the 3D printer. Next is, prosthetic manufacturers also have changed their approach to designing artificial limbs. Now many are able to print Fairlink prosthetic limb covers that mold perfectly to a person's anatomy, giving the wearer a more comfortable than we can think. So scientists have also successfully printed cartilaginous structures such as ears and tracheas, etc. to make the former bioengineers like 3D scan of the patient's ear, design a mold using computer aided software and then printed it out. So they inject the mold with cartilage cells and collagen. So after spending some time in the incubator, the ear comes out ready for attachment of the patient. Next one is, so 3D organs will play an important role in education and also in drug development. So it might even a factor into development of food and clothing products, lab gown, knit, something like that related to lab materials. So some having an access to such lifelike tissues also benefits pharmaceutical companies which can test candidate drugs to see their effects. So this is some of the basic uses with the help of 3D organs. This is about the general overview of 3D bioprinting. So you can approach us through these social media healthcare engineering platforms, and also if you want to know more content regarding this article, you can visit our blog. If you want any specific topic to learn, you can mention it below the comment section. Kindly subscribe our channel, and thank you so much for watching our video.